Hey, biology students, this is Dr. B, and we are starting part 3 of Lab 2.1, Meiosis in Gizmos. Remember, we're working with Calyx the cat. He has some kind of genetic disorder that makes him calico, and most male cats cannot be calico. They cannot be orange and black. They can be orange, they can be black, they can be orange and white, they can be black and white, but they can't be orange and black because they don't get 2 X chromosomes, and the gene for fur color is on the X chromosome. Since males only get 1, Calyx can only have 1 color, plus he can have white patches that's on a different chromosome. So we talked about number 15 the last time. There is the key concept that you need to fill in the blanks. Number 16 says, how many times must the germ cell split in half to create 4 gametes? Remember, we're in meiosis, we're creating gametes, that's eggs and sperm, that only have 1 copy of the chromosomes. So when they get together, they have 2. Egg plus sperm equals 2 copies of the chromosome. They're going to get together, make 1 cell, and that's the beginning of every organism on Earth. Pretty cool, huh? You and I and frogs and cats and everything else started from 1 cell. Eggs plus sperm. So let's continue on in meiosis here. We're in interphase in the handbook under meiosis 1. How many times must the germ cell split in half to create 4 gametes? In this case, in meiosis, it has to be twice. There's 2 splits. The first split makes 2, and then that split makes 4. So the 2 divisions are called meiosis 1 and meiosis 2. That's 2 splits. Meiosis 1 and 2 are very similar. They have the same 5 phases. Let's look at the phases of meiosis 1. Now, it's important when we look at meiosis, it's not just mitosis twice. The chromosomes line up a little bit differently, and let's pay attention to that as we go through here. So, prophase begins with the chromatids becoming more compact. They're shrinking up so they don't get all tangled when we line them up to divide them. This process is called condensation. It makes them smaller so they can be moved without getting tangled, because a chromosome, if it's all untangled, might kind of look like this. You kind of coil them up in a process called condensation, so they're a little bit easier to move around. This is easier to move than this, because there's lots of moving and sorting. The chromatids from the mother and father bind to make a structure with 4 chromatids called a tetrad. Tetrad means 4. So there's the tetrad. Now this is really important, and this is a big difference from mitosis. Mitosis, you don't get tetrads. Just the single chromosomes line up on the metaphase plate, and crossing over, which we're about to look at, where the 2 chromosomes exchange DNA, they switch parts, that crossing over, which results in variation, genetic differences among individuals, new combinations of alleles, that's pretty cool, and it creates unique chromosomes, combinations of alleles that have never existed before. So let's watch crossing over. They exchange DNA, so part of 1 chromosome goes to the other. They switch equal parts like this in normal cases. So here's the paternal chromatid not mixed up. Here's the maternal chromatid not mixed up. And here's the ones in the middle that have maternal and paternal alleles. So if this would be Calyx, this would be Calyx's mom's and dad's DNA in the same chromatode, and here as well. This would just be Calyx's dad's DNA, and this would just be Calyx's mom's DNA if Calyx were doing meiosis. This exchange of DNA is random, and crossing over can produce many different allele combinations. So every egg or every sperm is going to be completely unique. That's pretty cool. Usually there's an equal amount of DNA that crosses over. Sometimes there's an unequal amount, and that's where we start to see problems. When this happens, one chromosome gains genes and the other chromosome loses genes. So in a situation like this, where these two chromosomes are crossing over, they've switched some stuff. If there is a problem here, some of the material from one chromosome is going to end up on the other chromosome, is going to be added, and the other chromosome is going to kind of get gypped here. You can see how one chromosome doesn't have quite as much DNA as the other one. This one has extra, this one doesn't have as much DNA, and that happens sometimes. It's called chromosomal rearrangement. So let's watch that happen here. Part of a chromosome gets stuck on the other one. It's not an even exchange, and one ends up with more and one ends up with less. Here the genes have been rearranged by the chromosomes. This is called chromosomal rearrangement. And sometimes, a lot of times you already have one copy of the gene, so it's fine. But if you gain extra copies of a gene, sometimes it can cause problems. The X and Y chromosome have different genes, so they don't cross over. X and Y stay separate. If you're a female and you have two X's, they will cross over because they have the same genes. After the chromosome crosses over, organelles called centrosomes now move to opposite sides of the cell. You can see the centrosomes right here. And they're going to start organizing. They're going to send out spindle fibers. They're going to break the nucleus, and they're going to start attaching to all the tetrads here, those groups of two chromosomes hanging out together, and they're going to start pulling them apart. Now, in a human, you're going to have a lot of these tetrads, and it's a lot of things to organize in a tiny little cell. That's why you need the centromeres and you need the spindle fibers. In metaphase, the spindle fibers light up and the tetrads in the middle of the cell. So what we have here, basically, just to give you an idea of what's going on here, we have 1, 2, 3, 4 chromatids all lined up, and they're going to get pulled apart to 2 and 2 like this. So let's move our tetrads. So right now we're 4N. We have 4 copies. We're going to move everything over. And there's a bunch of randomness here. There's different ways that those chromosomes could get split up when the cell pulls them apart. And so that's part of the genetic variability here. There's random assortment here. Anaphase separates the tetrads and guides homologous chromosomes to opposite sides of the cell. Now this blue one could have been on the other side, this blue one could have been on the other side. There's a lot of different options that could have happened. The random lineup of tetrads makes the set of chromosomes that end up on each side random. Since the chromosomes can be pulled apart in 2 directions and cats have 19 chromosome pairs, anaphase can produce over 520,000 chromosomal combinations. That's pretty mind-blowing, and it's even more in humans because we have more chromosomes. This is very important for genetic variation in cats and in humans and in everything else that does sexual reproduction. In telophase, the spindle fibers break down Things get pulled over to their own side. We now are back to chromosomes, not tetrads. And that's something that's different than mitosis. Get the cleavage furrow. And now you're 2N. And now we have to get those 2N cells down to 1N, one copy of each chromosome. We've got cytokinesis. Now we've done our first separation in meiosis. Now we have to go to meiosis 2 and do another separation so we get down to 1N. Right now, all of these are 2N, and we have to get down to a situation where we have 4 cells that are 1N each. So each cell will divide again to make 2 new daughter cells. Mitosis 2 includes the same phases, but with 2 important differences. There's no interphase, so you don't double your DNA. Crossing over doesn't happen in prophase 2. So here's prophase 2. The spindle fibers come over to organize all the chromosomes here. Here's metaphase. We've got everything lined up in the middle. Meta means middle. Anaphase, we start separating our chromosomes. And notice there's only 1 copy here. And this is another place where randomness, the pink one could have gone over here. The pink one with blue could have gone over here. There's lots of opportunities for uniqueness and variation and randomness here. The difference in telophase 2 is that the chromosomes become uncondensed. They go back to their longer shape. And so now the cells can just go back to being cells. They don't have to be all ready for division. And those chromosomes can kind of unwind and go back to being kind of their normal chromosomal cells. Meiosis 1 and meiosis 2 results in 4 haploid cells. That means they each only have 1 chromosome. And they might have been mixed up. So now we do cytokinesis. We get 4 separate cells. We started with 1 germ cell, a diploid cell. Meiosis 1 and meiosis 2 resulted in 4 gametes. That's pretty cool. That's how we make eggs and sperm. And cats, and hippopotamuses, and zebras, and whatever other animal you can think of. Meiosis 1 and meiosis 2 contribute to genetic variation. Use the diagram to explain your answer. Well, remember we talked about how those chromosomes, the tetrads in meiosis 1, those 4 chromosomes all together, lining up at the metaphase plate. And that's an opportunity for them to get pulled apart. And we don't know which pair is going to go to which side. And then in meiosis 2, again, you have a chromosome pair. And we don't know which chromatid is going to go to which side. So there's lots of opportunity for randomness. There's that opportunity for crossover as well in prophase 1. So hopefully you have a good idea of how to answer this one. I'm just going to type some garbage in here. Don't copy this as your answer, so I can submit it and you can come up with your own answer. So, key concept, every gamete is genetically unique because of crossing over in prophase 1 and the random lineup of chromosomes in metaphase 1 and metaphase 2. So that's in meiosis 1 and meiosis 2. And then the separation in anaphase 1 and anaphase 2. So let's go back to our lab sheet here. We know this. How many times must the germ cell split in half to create 4 gametes? We know it's 2 separations. There's meiosis 1 and meiosis 2. What happens to genes in the maternal and paternal alleles in the tetrad? Well, this screenshot gives you a hint here. Those tetrads give the opportunity to create new chromosomes for maternal and paternal chromosomes to get mixed up, and they can exchange some DNA here, the ones in the middle. There's another one here on the outside, and another maternal chromosome on the outside there. The ones on the inside can cross over and exchange some genetic material here. You can see how the pink and the blue have switched over. And that creates new combinations of alleles, and it's pretty cool. What happens in chromosomal rearrangement? Well, the chromosomes don't come apart quite the way they're supposed to. One chromosome gets kind of gypped. It gives away some of itself, and the other chromosome kind of gets a bonus. They don't separate the way they're supposed to from crossover. Here's the chromosome with a bonus. Here's the chromosome that got gypped. It's missing this part that went over to the other chromosome. It's not entirely uncommon in humans or cats. The key concept you need is right actually in the screenshot there. The random lineup of tetrads makes the set of chromosomes end up on each side, and you can read the rest right there. You can blow it up on your iPad or your device if you need some help reading that. Why is there no interphase? I happily wrote an answer here for you unintentionally, but you don't want to double your chromosomes, because the whole point of meiosis is get down to one copy. So you go from 4, then you do meiosis 1 and you're in 2 in each cell, and then you go to 1 copy in 4 cells. If you doubled, you'd still have 2N, or 2 copies of each chromosome in each cell, and that would be trouble, because 2 plus 2 is 4 when you combine eggs and sperm, and that's more than you need in a cell. So how do meiosis 1 and meiosis 2 contribute to genetic variation? Prophase 1, remember there's crossover. Metaphase 1, there's independent assortment. There's also independent assortment in metaphase 2. There's lots of opportunities to not only mix up those chromosomes and get new chromosomes, new combinations of alleles, same genes, different alleles. So you get new chromosomes, unique chromosomes. These are still both like chromosome 3, but they're new combinations of alleles. That's crossover, and how these get separated into the different germs, into the different gametes, is random. So that's another opportunity for variability. And that's why sexual reproduction is so good for, so good at creating variability. It's got all these opportunities in meiosis for creating variability. I believe we went over these. Each gamete is genetically unique because of crossing over in prophase and the random lineup of chromosomes in metaphase 1 and 2. The male gametes can either have an X or a Y chromosome. This means that the chromosome in the male determines the sex of the child, and we're going to go see that here in one moment. Fertilization is the next big event here. We've made our gametes, we've made our eggs, we've made our sperm. Sperm, of course, in males, gametes in females. Gamete means husband or wife and comes from the word gamos, which means marriage. Fertilization is the marriage of sperm and egg. Cats, of course, don't get married. They just put sperm and eggs together and make kittens. In fertilization, the nucleus of the sperm cell fuses with the nucleus of the egg cell. So they're going from 2 cells to 1 cell. They're going from 1 copy of the chromosomes, 1 copy of the chromosomes, to 1 cell with 2 copies. That's the important thing about fertilization. So when a sperm is allowed into an egg, the egg makes the decision which sperm to allow in. The tail drops off, and just basically the nucleus of the sperm is in there. So you get all your organelles from your mom. So when they look at sort of your deep ancestral heritage, they look at your mitochondrial DNA. The DNA that's in your mitochondria, which you only get from your mother. The male gamete's sperm can either have an X or a Y chromosome. This means that the chromosome in the sperm that determines the sex of the child. So the father either contributes a Y or an X. Mom always contributes an X. So if you get an X from dad and an X from mom, you're female. If you get an X from mom and a Y from dad, you're male. There are lots of different things that can happen where you get two X's and a Y, or you get some combination of X's and Y's that isn't what usually happens, and you can have a condition what's called intersex. You can certainly look that up online. It's now a legal definition. You can be intersex on your passport if you are not clearly male or female. That is now a legal option for folks who end up in that category. But most people end up with XY or XX. And they both function, and they're either very clearly female or very clearly male. If you don't fall into either of those categories clearly, that's okay. That's cool. And of course, your gender, you get to choose. Your chromosomes, you don't. Which of the following is true? Fertilization requires two haploid cells. Homologous chromosomes have the same alleles, but they may have different genes. Nope, other way around. Same genes, different alleles. So that's not true. Diploid cells have two identical copies of each chromosome. Nope, they've been mixed up. Oh, wait. Diploid cells have two yes, that's true. I know this one's true. It should be A and C. Okay. Oh, they're not identical. One's from mom and one's from dad. So they're homologous, but not identical. When they're copied, you have two from mom, two from dad right before the cell divides in mitosis. Fertilization brings together two haploid gametes. That's half as many chromosomes. That's one N, or one copy of the chromosomes, and an egg or a sperm to produce a diploid. That's two copies of the chromosome, zygote. A zygote is like the first cell we make organisms out of. It's egg plus sperm, and you start making the whole organism out of that single cell. All the organisms that do sexual reproduction and don't lay eggs come from that one diploid cell that's fertilized, egg plus sperm. Egg laying is a little bit of a different condition, but cats, dogs, people, not alligators, giraffes, horses, all of those organisms come from one cell, and it grows and grows and grows and makes something bigger and amazing. The zygote divides by mitosis. That's creating identical daughter cells. Mitosis is very similar to meiosis too, and it's also broken down into prophase, metaphase, anaphase, telophase, and cytokinesis. So now we're just making exact copies. We have the zygote, we have the beginning of a new organism, and we're just making more cells to make more organism. Because you're a couple of trillion cells. You started from one, but now you're a couple trillion. And so you had to make a lot more cells to be as big as you are. DNA mutations can also happen in meiosis or mitosis when the DNA is copied incorrectly. When this happens in meiosis, it can change the genotype of the offspring. DNA mutations in mitosis do not change the genotype of the offspring. So if mutations happen in meiosis, you can get a genotype difference. If it happens in mitosis, like you get a little bit of a genetic difference in one of your skin cells, it can be skin cancer, but it's not going to be all of your cells in your whole body. It's going to be a few cells on maybe your cheek. Crossing over does not occur in mitosis. This is how the daughter cells stay genetically identical. So when you're doing mitosis, I think of it as happening in mitose, and it's pretty boring. It's just making more cells that are exactly the same. So calico cats have orange and black fur with white patches. Let's investigate the two genes that cause this coloring. So the white spotting is caused by the S gene. S stands for spotting, which has two alleles, big S for spotting and little s for no spotting. The big S is called dominant, so if there's at least one big S, you have spots. Calyx has white spots, so he's definitely got one big S allele. So that's separate from the orange and black. The rest of the fur color is controlled by the O gene that has two alleles. Big O is orange fur and little o is black fur. So this is a cat with orange fur. This is a cat with black fur. The O gene, or the fur black or orange gene, is on the X chromosome. Males only have one X chromosome, so they only have two options for colors. They can either be orange or black. They can also have white spots, that's on a different gene, but they can't be orange and black because they only get the orange gene or the black gene, and then they can have white spots or not. They can't be orange and black because they only have one chromosome, and that is where we will stop. So, which of the following is true? We clicked on A. Explain why a zygote is genetically unique. It's got genetically unique chromosomes. It's gone through two processes of splitting those chromosomes randomly. Genetic mutations in meiosis do change the genotype because everything gets made from that original cell in meiosis. But in mitosis, if you're doing mitosis of your skin cells and your skin makes an error in copying the DNA, that's not going to affect your whole body. It's going to affect a little patch of skin cells. What does the word dominant mean in this context? Well, hopefully you remember dominant versus recessive. And take a good look at this diagram and try to wrap your head around it before you get to the next video, because this diagram is explaining what I was talking about. Only females who get two X's can have a big O and a little o, can be orange and black. Males can be black because they get one X, or they can be orange, they get the big O on their X. They don't have a second X. Females can be orange and be all orange. Females can get little o and be all black, or females and only females can get big O, little o and be black and orange. But males can't because males only get one X chromosome. I'll see you next time to finish up.